Hello, everyone. I'm Tiff Bortwood, and this is DNA Dialogue, a virtual forum to learn of new developments in health genomics and engage with global experts in the field. As we come together today in the spirit of reconciliation, I acknowledge the traditional custodians of country throughout Australia and their connections to land, sea, and community. I'm joining you from the lands of the Wurundjeri Woiwurrung people of the Kulin Nation. I pay my respects to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples joining us today. Please ask any questions you have using the Q&A function during the presentation. I'll do my best to get to as many of them as I can during the Q&A time at the end of the talk. And if you're active on social media, we encourage you to use the hashtag DNA Dialogue and please tag at AusGenomics. So today we're joined by Dr. Euan Ashley, who is the chair of the Department of Medicine and Arthur L. Bloomfield Professor of Medicine and Professor of Genetics, of Biomedical Data Science and Pathology at Stanford University. He's best known for his contributions to the field of medical genomics. He's the founding director of Stanford's Clinical Genomics Program, the Centre for Inherited Cardiovascular Disease, and the Catalyst Program for Biomedical Innovation. He's decorated with many esteemed awards and has co-founded 5 biotechnology companies. His book, The Genome Odyssey, Medical Mysteries and the Incredible Quest to Solve Them, was released in 2021. In his presentation today of the same name, Euan will contextualise the breakthroughs of precision medicine through the real diagnostic journeys of his patients and the efforts of his fellow doctors and scientists as they hunt to prevent, predict and beat disease.
All right, well, I'll get started. Yeah, thank you for the kind introduction and thank you for the invitation to present today. It's really a pleasure and a privilege. For me, it is the afternoon of Wednesday. I believe it is Thursday morning for you, and it's great to be virtually in Australia. It would be great. It would be better to be actually in Australia, but this will have to do for now. As Tiff mentioned, I have some financial disclosures, which are shown here on the screen. In the recent past, I have also been including at the bequest of Nature non-financial disclosures, which actually I think for most academics are probably larger conflicts. And so here are my institutional loyalties, personal beliefs, ambitions, and other influences. As you can see on the screen, I've basically spent many years focused on genomic medicine and artificial intelligence. And I thank you for asking me to talk about genomics, since most of my recent invitations have been to talk about artificial intelligence. So it's great to dig a little into the archives and tell you a little about the work we've been doing over the last 10 years. Of course, the genome is the thing that brings us all together. It is the incredible ability of our ability to read the genome that has changed so much for patients with rare disease and increasingly patients with all forms of the disease. And as a cardiologist who spends a lot of my time looking at cholesterol panels with 5 numbers on them, then the 6 billion letters of the genome, 3 billion base pairs and 23 pairs of chromosomes. This is data on a completely different scale. I usually include this at the beginning of many of my talks, and I tell the students and others if I'm giving a talk to the lay public that if you unraveled all the DNA of every cell in your body, it would stretch to the moon and back thousands of times. But I also made a slide to try and visualize for myself what kind of size of data that is. And this is actually the myosin heavy chain gene. But if you were to stare at this screen going forward, it's probably a little blocky with the internet connection. But it would take you 3 months before you saw your entire genome if all you did was stare at the screen and it scrolled continuously at the speed it just did a moment ago. So the first genome, or at least the first half genome, or at least the first half of 92% of a genome, was released in June 2000. And Francis Collins and Craig Venter are here, Bill Clinton just off the screen. I think Tony Blair was somewhere too. This was a multinational, of course, 10-year program funded at $3 billion to try to sequence a human genome for the first time. And like many genomicists, I'm sure including many of you in the audience, you've been showing for many years this particular graph. Because we're from Silicon Valley, we love things that relate to computers. So we love the fact it's a Moore's law graph. But actually, I was in a a meeting several years ago with an Australian genomicist, Daniel MacArthur, and he used to like call out people on social media for showing this graph because everybody was showing it. And he would have a bingo card for genomics things. And I realized at the time I was sitting in the audience feeling a bit nervous, thinking I needed something else that people could relate to that would give a sense of just how much a genome had reduced in cost. And, you know, Craig Venter's genome, 100 million, potentially $100 now, depending on the technology and how many you sequence at the same time. Of course, you can't get $100 cost on just one. But I thought of something else, and I'd been driving past this car garage on the way home from work, and I realized that this car was available. I'd been doing the mental math, and at the time, it cost about 400,000 US dollars, a little bit beyond the budget of you know, a lowly assistant professor at Stanford. But as I calculated how much the Ferrari would have dropped in cost, had it dropped in cost as much as human genome sequencing had dropped in cost, I realized it would be 40 cents, which, well, that's a little bit more affordable. And I realized after I used this analogy in place of the Moore's Law graph that I just showed you, this was all anyone remembered about my talks from then on. And people would come up to me at meetings afterwards and say, hey, you're the guy with the Ferrari slide, aren't you? Sometimes they wouldn't even remember what kind of car it was. I'm happy they took something away from my talk, but maybe that wasn't the most important thing. Over the years, I updated the slide. For a while, it was 10 cents. And then for quite a period of time, it was 5 cents. And then just over the last year or two, based on that $100 price, we have a 1 cent Ferrari, which is a pretty wild and crazy thing when you think about the potential for good. And I hope to outline over the next half an hour for you some of the examples of that. For us, though, the story really began in 2009. This is a colleague and friend. I didn't know him at the time very well, Steve Quigg. He became the 5th person in the world ever to have sequenced his own genome. In fact, in his case, with 2 people in his lab in 5 days for about $40,000. Now, I've just told you can have a $100 genome. I'm sure some of you have availed yourselves of at least cheap genomes in the hundreds of dollars sort of price. 40,000 sounds like a lot, but remember the one before that, the 4th one, cost around $200,000 or $300,000. So this was a very significant drop in cost. And the fact that he did it in his lab in 5 days with just 2 other people, not a large multinational consortium, was what was remarkable and led to these kind of headlines. And I actually went to meet with him, not about this, but about something completely different. But he pulled me over to his desk at the time and said, hey, come and look at this. And on the screen, he started showing me a list of variants. And this is not something that I or really any other cardiologists had seen, even though I ran a Center for Inherited Cardiovascular Disease. I'll tell you a little bit more about that in a while. But we were sending gene panels of 3 genes, 7 genes, 9 genes, this kind of thing. And it would take 3 months to come back, and it would cost about $5,000. And here he was suddenly showing me variant lists that were his entire genome. And that was kind of mind-boggling at the time. But then he also started pointing at actual genes like myosin binding protein C, a cause of hypertrophic cardiomyopathy, and said, hey, look, what do you think of that variant? And I was impressed. He thought I had a photographic memory for genetic variants. Of course, I did not. But I did recognize that gene, and it is a cause of hypertrophic cardiomyopathy. So I started asking him about his family history of heart disease. And over the course of a few minutes, he laid that out. And it looks a bit like this. Everywhere you see orange, there's some form of heart disease in his family. You can see, of course, on his father's side, quite a significant history. And given that he was talking about diseases that can be associated with sudden death in young people, I asked him if there was any sudden death in any young people in his family. And he mentioned his cousin's son, shown here, a 19-year-old who had no prior medical history and just simply died in his bed one day, really tragic. I realized at that point that he was about to become the first patient in the world to walk into a clinic because I was in the middle of inviting him to my clinic because clearly he needed to be screened as a middle-aged man with this kind of family history. And he was going to show up for screening with his genome on a hard drive. And that was a little scary because we had no idea what to do with the genome at that time. And so what do you do when you have that kind of problem? Well, you call some friends, and that's what I did at Stanford, and initially some folks in my lab. I tried to find photos of them from this era, so they look like I do, much older now. But this is photos from their era. Matt Wheeler is now a faculty member here at Stanford. Joe Dudley and Alex Morgan are both venture capitalists, of course. And Rong Chen is at Mount Sinai. They were from the labs of Atul Butte and Russ Altman. And along with Steve Quake's lab and an ethics group, we put together a framework. It's actually many parts of it still around as part of the Stanford framework for interpreting human genomes all these many years later. But it was really a sprint, a kind of 6-month sprint between when we first saw the data and when we published this paper. And those were pretty heady days. This was really early in genomes. There were no genomes available, or no exomes even available clinically at the time. So this was something that we were happy with, but it was very embryonic. It was a very early days. But it turned out that there was, in the end, quite a lot of attention. We were, for a viral moment, it wasn't called viral moments at the time, but we were viral for just one moment, a very short period, where we got calls from all sorts of newspapers and TV shows and documentary producers, and even we had an exhibit at the Smithsonian for a little while. So we got some attention, I think, just for the sense that people think of their genome, probably wrongly, as, you know, potentially a little bit like destiny, or at least as something that can say something about their future health and their future risk of disease. And that caught, I think, their imagination in the way that they thought was newsworthy. And for us, it was the start of really a decade of trying to think about how to do genomics clinically in a safe and cautious way, how to push genome sequencing to be of greater quality, to move it from kind of discovery quality to a point that it could be real clinical quality. And so many people in so many countries around the world were participating in this, and our contribution was only small, but we were happy to be a voice as part of that chorus in thinking about how to move this forward. The first patients, of course, who benefit are rare disease patients, and sometimes time is of the essence. This was one of the earliest patients that we diagnosed using a rapid turnaround approach. A little baby who was found to have what we thought was heart block initially when she was still in utero, so about 36 weeks of gestation, and was airlifted to Stanford Children's Hospital, where she underwent cesarean section in order to have this ECG performed, which showed very long QT. So long, in fact, that the next beat was blocked, which is why she ended up with heart block. But that by itself isn't a problem. Like a low heart rate of 60 or 70 beats per minute isn't necessarily an issue for a little baby's compliant heart. But this is a problem. This is torsade de pointe. This is ventricular fibrillation, cardiac arrest, rhythm. And in the first day of her life, she had suffered 5 cardiac arrests and had CPR 5 times in the NICU, the neonatal intensive care unit at Stanford. So we had to do something beyond CPR. We implanted an ICD. This is it shown, an implantable defibrillator shown here in her abdomen. In adults, they're put up here under the collarbone, clavicle, but in little babies, and she, I think, was maybe even officially the youngest baby ever to have had one of these defibrillators implanted surgically on the first, I think, 3rd day of her life in the outside world. But more important than that was, could we help? We were throwing everything at the wall to try to save her. And this is with long QTs, one of these things where you just literally throw every drug that you have. But if you can understand what the genetic basis of the disease is, you can actually do precision therapy. You can take away some things that might be harmful, and you can emphasize the therapeutics that might be beneficial. So at the time, we had no real mechanism of doing rapid testing, but the lab at Illumina had been working with Stephen Kingsmore, who at the time was in Kansas, now in Rady Children's, to try to speed this up. And he had published a protocol around this time, 56 hours. And we thought that there were probably ways that computationally we could shave some minutes off that. And that task fell to James Priest, who's one of, at the time, was a postdoc in my lab, pediatric genomicist and cardiologist. And he spent the weekend basically revising our algorithms ready to receive the data, and ran this kind of as soon as it came back, which was over a weekend. And we found essentially 2 variants, one known variant in KCNH2, a potassium channel gene known to cause long QT, and then another in this gene RNF207, which wasn't known to cause Mendelian long QT, but was found in multiple GWAS genome-wide association studies. And this was a frame shift in that. And one was paternally inherited, one was maternally inherited. Neither parent was obviously affected. So we were looking for a de novo or a compound het or a homozygous mechanism here. So it seemed like we likely had the answer, and were able to relay that directly to the bedside team, who were able then to alter the precision therapies, but knowing this was now a potassium channel form of long QT, not a sodium channel form of long QT. And could actually withdraw one of the therapies that has a pretty narrow toxicity window. Interestingly, it took us probably, and I say us, this is very much collaborative work, mostly driven by a collaborating lab in the University of California, Davis University. But if you look at the top publication here, 2014 is when this first paper was published, the genome paper. This graph here is from the second heart rhythm paper, which was published 8 years later in 2022. It took that long to finally put the mechanism together. And this is the thing about genomics. Some things can happen fast. You can sequence a genome fast. But in this case, we had a novel mechanism. We were fairly sure about this variant, but inherited by the father who did not have long QT. Clearly, this was not the whole story. But the whole story was laid out by our collaborators, Nipaban Chiamvunmat from UC Davis. And shown here are whole cell patch clamp recordings of the potassium current. First of all, shown with the wild type current here, the hERG current, it's the KCNH2 potassium repolarization current, RNF207 wild type. So these are normal whole current density plots for the current of the potassium repolarizing current. Here we have the wild type hERG with the abnormal, sorry, the heterozygous hERG with the wild type RNF207. And you can see in red, basically very little change. So this is the situation with the father shown here. This is his, and this explains why his long QT is essentially normal. But when you have the two variants together expressed heterologically, then you have the blue, which is a very significant change, as you can see in the current density. But it took 8 years to get there. It's obviously possible to do this faster, but the mechanistic science that sometimes follows the discoveries we make takes a long time, and often our funding bodies don't realize just how long that could take. But I was happy to say that after we managed to make more precise her therapy during that inpatient stay, she was discharged. This was a picture of her taken 6 months later. Kathleen, little baby, now a little girl, but actually this was more than 5 years ago. I need an updated picture. I actually got a picture when she was 5 years old, but she has done very well, still has the defibrillator, but has stayed mostly out of hospital and away. And I think still to this day probably is somewhat unsure of the amount of technology that was thrown at her early days to keep her alive, but she's been doing well. So in her case, we knew the answer. We knew what part of the answer. We didn't know the molecular answer, but we had a diagnosis in the sense that it was a clinical diagnosis. We knew it was a long QT. There were some things we could do. But for many patients, they go from doctor to doctor, from provider to provider, asking questions, describing symptoms without any answers. And for those patients, they're often referred to as being on a medical odyssey. And so, of course, is after, and they accumulate paperwork, just like this is a real example shown here. Piles of paperwork, piles in the US at least of financial burden as well as the emotional burden. And they are undiagnosed. The patients often describe that they're on an undiagnosed island. They feel like they're on an island because they don't know what they have. They don't have a community of people who share the same condition. They don't have any idea how they might treat it or what might happen. And of course, that journey they're on is often called a medical odyssey after Homer's Odyssey. And I forgive you if you haven't read the entire book recently, but it describes Odysseus, the king of Ithaca, and his 20-year journey to get back to his wife and his betrothed, fighting off one-eyed giants who eat his crew, a variety of shipwrecks, hypnotic siren songs of beautiful women trying to encourage shipwrecks again. Monsters that have dog's head and shark's teeth. It's pretty gory stuff, and actually probably isn't a bad analogy for what many of our patients and their families go through. I pulled this Homer's Odyssey a few years ago, put it on a slide. It took me a few times of showing it before I realized under here it says one of the 10 best books of 1996, which I thought was quite interesting. Even more than that, I pulled it from Amazon, so it actually gave you the option of following the author just in case Homer publishes any more books. I've been waiting a while, and it turns out Homer hasn't published any more books, but I live in hope. The Undiagnosed Diseases Network is focused on trying to solve those kind of medical mysteries, and it grew out of the Undiagnosed Diseases program that was at the NIH and is still at the NIH. And the Undiagnosed Diseases Network is spread across the US. It's connected internationally to UDNI, the International Undiagnosed Diseases Network. And the idea is to apply genomics and really the cutting edge of clinical medical care to the most difficult cases in medicine, the people with no answers, and to try not just and find them answers, but potentially find them treatments. And one of the reasons I particularly love being involved with this network is it combines traditional approaches to medicine. It's very much connected to the history of medicine. It's kind of the Sherlock Holmes approach, where we gather the clues, we think carefully, we put experts in the room, we examine the patient, we talk to them about their history, and try and work out what's wrong. And 10% of the time we can solve cases just by doing that. And that's important. But that's not enough, because that leaves 90% of people with cases unsolved. And it turns out that genome sequencing is the single most powerful technology for solving those kinds of cases. Now, there's other technologies, and we use all of the ones shown on the screen, but the majority, which say about 3/4 of the cases that are solved, and is about a 30 to 40% solve rate overall, and 3/4 of those solved cases are solved by genome sequencing. So I wanted to tell you one story of a family. This was featured in Stanford magazine about Carson and Chase Miller. Carson is on the right and Chase on the left. And Carson was born first. Their mom and dad were called Danny and Nikki, and Carson hit all his milestones through the first year, basically. And by a year, those of your parents know that by a year, kids are kind of standing up a bit, usually cruising around furniture. They can hold implements and feed themselves a bit and start to talk a little as well. And he'd hit all of those milestones until he reached a year old, when he started to slip back. He didn't slip back cognitively. It would seem like his intellect was intact and progressing, but motor skills got worse. He was no longer cruising. He was on the floor. And it got to the point where in the end, after a year or two, he needed to be in a wheelchair, as he's shown here. And then his younger brother was born. I mean, they had by this time taken Carson to multiple doctors and had no answers. He had a label of cerebral palsy, but that doesn't really mean anything, and certainly doesn't point at any potential answer. And then Chase, his little brother, went through exactly the same thing, hit all his milestones, and then about a year old started to slip back. And just to put you in their world for a moment, you know, their father, Danny, worked full-time. Nikki was at home full-time on the kids. Of course, they're both now in wheelchairs at this point. They've been going to doctor's appointments, trying to find answers, pediatricians, neurologists, developmental specialists, endocrinologists, another neurologist, and they just have no answers. And meanwhile, their daily world is she has to help both of them get up, both of them get dressed, both of them eat their meal in the morning. Danny goes off to work. She takes them about during the day and has to help them with essentially everything. And then Danny comes home at the end of the day, exhausted from work, but she's exhausted from her time spent helping the kids. So he would take over, and then she'd go to bed, and then he'd put them to bed, and then he would go spend 4 hours, 5 hours on the internet trying to find an answer. He was just sure that somewhere in the vast expanse of the internet, he must be able to find the answer. But so far at this point in time, no doctors had been able to find the answer for him, and they'd had by this time quite a lot of testing. Some of it is shown here. They had brain MRIs, which was really the only thing that was positive, in the sense that there was a finding in the basal ganglia that pointed to the symptom basically of the motor dysfunction, but it certainly didn't explain anything. And none of the other biochemical tests were anything other than normal. There was an exome done individually on each boy, and that didn't find anything either. So they'd had pretty significant genomic testing at the point that they called the Undiagnosed Diseases Network, and one of their neurologists sent them to the network, and they were referred to the Stanford Center because they lived locally here in the Bay Area in California. And we standardly, as part of the admission into the Undiagnosed Diseases Network, do nuclear family sequencing. So that is usually a trio or a quartet. And in this case, we did all 4. And in this case, the answer basically popped out. Which is to say, this gene, mitochondrial trans-2-enol-CoA reductase, MECR for short, was found to harbor compound heterozygous variants, the same variant in each boy, each one passed by one parent, and shown here, the mother's T insertion, shown here, inherited by both siblings, and missed completely by the exome because it didn't include the 5' UTR, was this variant here, the sense variant, passed on from the father to the 2 siblings. This made the boys the 8th and 9th individuals, 8th and 9th kids in the world to be diagnosed with MEPAN syndrome. And that's not a syndrome like most of the rare disease long tail that any of us had heard of before. And I'll forgive you if you're not that familiar with the mitochondrial FAST2 pathway, which is a fatty acid synthesis pathway. There'll be no testing at the end. I don't think I ever learned this pathway, but there is indeed inside mitochondria a fatty acid synthesis pathway, and it looks like this. And the MECR gene, shown here, has a synthetic enzymatic function to produce acyl ACP that goes on to produce octanol ACP, which produces lipoic acid. The details don't matter. It's really to say that Danny suddenly had something he could look up because we had an answer now. I mean, it was a label. It's not a therapeutic. It's not any kind of miracle cure, but suddenly they moved from undiagnosed island to a place where they had a community. It was a small community at the time, only 7 other families. Now it's something like 30, 40, I think 50 families. But at the time, pretty small. But having an answer just meant that the size and shape of the enemy was now known. And more to the point, he was no longer kind of at sea in the entire expanse of the internet. He actually was able to pull up he put himself essentially in a biochemistry crash course. He pulled up this pathway and he started Googling the pieces in it. So maybe, well, what about this downstream stuff here? If this enzyme is missing in my boys, maybe I can give them some of this stuff. Maybe that's a way that I could help treat them. So he did that, basically Googled lipoic acid. And rather than, some of you will be familiar, but rather than find some incredibly expensive gene therapy that's $1 million or something, he found a supplement that was available quite literally on Amazon. So the genome sequencing had taken them to the point where he had no idea what was happening, no answers, no labels. They were on undiagnosed island to the point where they have a label, they have a community, and he can order something that can arrive through the mail the next day from Amazon that for $16 that might have some impact on his kids. And in fact, with the permission of his neurologist, he actually started giving that supplement to his kids and they started to improve. Again, it's not a miracle cure, it's not a gene therapy. They're still wheelchair bound, but they started to hit some of the milestones again and actually start to improve. Which is one of just many stories. And I think those of you who are involved in genomics, of course, you probably have your own, I'm sure you have your own versions of these kinds of stories. They're what keeps us driving forward. And Danny and the 2 boys were kind enough, along with Nikki, his wife, to be part of this CBS This Morning TV feature on the Undiagnosed Diseases Network when we published the paper. And this is now pretty old, actually, but this was just about 18 months, 20 months of the network. And in that time, we were able to define 31 new syndromes. Some of these numbers I mentioned already, 75% genome of the 35% diagnosis rate. And the actionability was 80%. So 80% of the time, and we have a whole framework for what actionability really means, because the reviewers were very focused on actionability. Actually, the patients are mostly interested in, do we have an answer? Because to them, that's the big change. Of course, they want a therapy. But the difference between being on undiagnosed island and being on diagnosed, being part of a diagnosed community is a major change. And it also means that they can now fundraise to go try to find a therapy. So, and Danny's done that. There's a foundation now for MePan. He's raised money, his research is working on it. The possibility of genetic therapies are not so far away anymore. Another really important and under-recognized element, I think, even probably undersold by our community, our genomics community, to the world is, look at this one on the bottom. Costs were 6% of price. So in this paper, we did a basic modeling of some undiagnosed patients who were in the same healthcare system before and after genome sequencing. We determined that if you'd applied genome sequencing earlier in their course, which don't wait till they've accumulated hundreds of thousands of dollars of diagnostic testing, most of which is redundant with prior testing, but do it early, you could save 94% of the healthcare costs. This is not just something that is cost effective. This is something that will save the healthcare system a huge amount of money. And yet, at least in the US, we still have healthcare payers who, for complicated reasons, aren't always willing to pay for this kind of testing. Maybe our proudest moment in terms of the press that covered this paper was this, because for a while, the news in the world looked so bleak that the New York Times started doing the Week in Good News. And we were 1 week the headline for the Week in Good News for the Undiagnosed Diseases Network. So I was pretty happy that we were able to be part of that. I mentioned earlier our Centre for Inherited Cardiovascular Disease. And in fact, this invitation, I think, originally came as an email from Jodie Ingalls, another Australian genomicist. I've had the pleasure of getting to know her over many years through our shared interest in inherited cardiovascular diseases and various meetings that we've had together as part of a group internationally who are really focused on these diseases. We set this up about 15 years ago at Stanford. And it's essentially an integrated center that looks after patients with these different diseases. Familial cardiomyopathy is our biggest single group, but we also have clinics in neuromuscular disease and familial lipid syndromes. I mentioned long QT earlier, also Marfan syndrome, aortopathies. And even sports cardiology actually falls underneath our umbrella because of the overlap between many of these things and changes that are normal for athletes. So we're able, thankfully, to see a number of athletes and rule them out for cardiomyopathy or long QT. And to give you a sense of how this has changed in the years, it was me and a half-time nurse in 2008. This is one of our patients in the middle, by the way. And in 2024, we're now somewhere like 15 MD clinics. There's a few other clinics run by our nurses and genetic counselors. We see 18 new families a week, and I won't read the rest of it for you, but it's been a growth area for us and a lot of impact because this was really an underserved community before. We, of course, do various conferences. We just had Ray Hirschberger talk at our conference about 2 hours ago. Some of you may know him if you know about the genetics of dilated cardiomyopathy. This is not from 2 hours ago. This is what our meeting looks like. At least it did a few years ago. We have run various fundraising events as well. We used to do a push-up competition with the 49ers football team, American football team. That's an old folk picture. It may even have me doing push-ups. I'm not sure. I'm counting. Look at me. I'm in the background, like not doing any push-ups here. But I did some. And then the last 2 years, we have a charity run that actually some celebrities from Silicon Valley, including Mark Zuckerberg, tend to come to every year. Runner's World even featured his surprisingly fast 5K that he did. I see it was raining at that point. It doesn't rain all that often in September, but that day it was running. I think these are his bodyguards who are running with him. I suppose they also have to run surprisingly fast 5Ks, is my guess. Anyway, the center is now run by Vicky Parikh, shown here and in the middle of this group photo. I am now leaning back in my armchair as the founding director while she does all the hard work. But it has been really a joy to look after these families and to be able to deploy genome sequencing through our clinical genomics programs, another group that we started a little later, 2014, I think. 3 years ago now, if I'm guessing correctly, 2021, we launched whole genome sequencing as a clinical service for these patients, which is to say that we have a 40 to 45x short read whole genome backbone that is the basis of all our genetic testing, germline genetic testing. And so we just essentially read out the cardiovascular panel or the renal panel or a variety of other panels, but what is banked for every patient is their entire genome. And it does mean that we can add polygenic risk scores to panel testing for modifying impact on Mendelian testing, and also add in pharmacogenomics. We actually have an independent pharmacogenomic program. Jack O'Sullivan is actually still a fellow in our program, and he led this polygenic risk scores statement from the American Heart Association. So we're interested in that. And here is our pharmacogenomics group. Stuart Scott is the director now of our clinical genomics program, something else I retired from. And Terry Klein, who for many years has run the PharmGKB knowledge base for pharmacogenomics. They have designed just in the last year this PacBio hi-fi sequencing through Twist that is actually available to anyone, so anyone can order it, but designed through a Stanford PacBio collaboration and rolling out early next year at Stanford Hospital. Will eventually be broadly. At the beginning, it'll be mostly in pain and neuropsychiatry applications for CYP2D6 predominantly. But this is reimbursed by Medicare, which is responsible. It's $1 trillion healthcare system responsible for the healthcare for those above 65 in the US. So it's actually something that, while no genomic testing makes much money, as far as I can tell, perhaps in cancer, then in the US context, then at least pharmacogenomics will have benefit while breaking even, similar to some of the other genomic testing that we have. And happy to share any details or connect to anybody who's interested in pharmacogenomics with Stuart and Terry. wanted to share one more thing about some of our work in the inherited cardiovascular disease space. We all maybe are familiar with a site frequency spectrum that looks a bit like this, long tail. This is it for hypertrophic cardiomyopathy and the population variants. This is also population variant graph from Gabor Marth, who is part of our UDN on Diagnostic Disease Network. This graph you might also be familiar with. As you test more genes, you find more variants of unknown significance. What is the answer to that? Well, there are many answers, none of them definitive, but one we hope to be a contributor is our newish consortium called Cardivar, led by a few of the folks shown here on this photo, and then also this is the first paper really from our consortium, led by Brendan Floyd and Vicky Parikh, but the PIs here also include myself and Dan Roden and Fritz Roth and Cal McCrae. And the idea is to produce variant effect maps. Basically, they would look like this. They would be a cellular model, in vitro model of every possible variant at every position across a gene. And this is the calmodulin gene. These genes can be associated with Mendelian syndromes that can cause sudden death. And so a typical scenario might be this one. Here's an 11-year-old that presented to our hospital with ventricular fibrillation and a long QT. The variant that was suggested to cause this, the calmodulin variant, was not found in any of the population databases. There was no useful family information, so it was classified as VUS. Both of the variants that were considered were in the map, which is shown here in the P80, the protein 80 position here, and 83. The darker blue, slightly lighter blue, but all on the likely pathogenic spectrum. So it's certainly never going to be the definitive answer, and probably a single lab, single assay is not going to be something we put a lot of faith in. But when multiple labs, as will happen shortly, start producing these variant effect maps, and where they agree with other data, let's say from population data or from increasingly good biophysical modeling or AlphaFold modeling, then we're I think going to get to a point where we have made some progress in the VUS problem. There's another example here that maybe for time I'll just leave you to read. But this is something that we're very interested in trying to improve genomic diagnostics, to think about cellular models as well as computational and AI-based models. So I wanted to share another last story with you here, and this is a patient called Matthew. He came to us from Oregon. In the first winter of COVID, he had a cough that his parents assumed was COVID, so they took him to the local hospital. He was admitted there actually because he looked pretty ill and diagnosed, in fact, with heart failure. This is his echocardiogram. I hope the video projects. We'll see if it does. But for those of you who might be used to looking at videos, there are probably not too many, but some videos of hearts beating. This is a heart that is not beating well at all, and is severe heart failure, and this valve here is leaking severely. He was basically in shock and had, in fact, to be airlifted to Stanford Children's Hospital, where he rapidly deteriorated in cardiogenic shock and had to be crashed onto extracorporeal membrane oxygenation, ECMO, which is a heart-lung machine. From there, he had a surgical implantation of a heart assist device called an LVAD, shown here. And this is him actually sitting outside the ICU. This battery is running his heart, essentially, at this point. He looks pretty pale. You couldn't really tell, but this is him actually outside the ICU. The question is, for a heart that looks this bad, how do you make a transplant decision? And it is possible there are some reversible causes of myocarditis that you could potentially diagnose by biopsy. Biopsies are rather barbaric, ancient, well, not that ancient, but feels like a barbaric ancient technique. But basically, you put a catheter in through the right ventricle, grab a piece of the septum here shown, and pull it out. Except you don't get this beautiful view here. What you get is this. So you don't really know where you are. And you open the catheter and grab and you pull. And then if you're lucky, you get mostly just a small piece of heart. Sometimes you get a piece of valve and other things. So it's not a great procedure. Maybe you see fibrosis like this. And in his case, it was really non-specific. You can rule in myocarditis if you see lots of white cells, but you can't rule it out. So the question is, was this a genetic cause or not? And was there a role for genome sequencing? Now, to take you back to the earlier case, I've mentioned Stephen Kingsmore and rapid sequencing. He had managed to do this 50-hour protocol. We had shaved that to 42. He had subsequently gone on to shave it down to 26 using Illumina, and by cutting some corners, I think, on the cluster growth of Illumina, you could get it even down as low as 19.5. This is not standard, I think, for that rapid turnaround. It's more like 30 to 40, but but 19 is possible, and he had been awarded a Guinness World Record for fastest genetic diagnosis, shown here. Of course, at Stanford, we're not competitive or anything, but we wondered if it might be possible to go faster. And if we could do that with newer technology, maybe we could even improve accuracy. So I put that question to these 4, at the time, PhD students. Now, 2 of them have graduated, 3 of them have graduated, these 3. Tanner is still in the lab. And to say, do you think we could put together a collaboration to see if we could go faster? And it would be based around this machine, this beast, Promethean, from Oxford Nanopore. And the idea was, could we literally take all the flow cells, 48, and put one genome on them, basically, and run it like full tilt, like pedal to the metal, foot to the floor. And you can produce a lot of data in a pretty short length of time. I mean, these are 4,000 channels, each with 3 nanopores. So you have about 12,000 nanopores per flow cell, about 500,000 nanopores per Promethean. So, and basically, for those of you who are, probably most of you quite familiar with nanopore sequencing, but you're, unlike growing a cluster with the sequencing by synthesis approach, you're actually unzipping the DNA, and essentially this motor protein shown here dictates how fast it then falls through this protein pore. The pore is embedded in a membrane across which there's a current, and that current changes according to the group of nucleotides that are currently inside the pore, and also their methylation status. And you get this squigglegram, which I think cutely they really do call a squigglegram. And you apply various deep learning and artificial intelligence approaches to that to call out ACs, Ts, and Gs, and methylation status. So we put together this collaboration with Google, with Stanford. You see it's industry and enterprise, it's tech, it's biotech, and also people who make processors and GPUs, and really tried to reinvent the process from beginning to end to make it faster. I'm going to just take you through Matthew's timings, or this is, I think, the first slide is a summary of the approach, so I won't go into it in detail because of time. But I will take you through him quickly. This is his actual genome with the time. We start with a couple of mils of his blood. This is actually not the fastest protocol for extraction of fragmentation. It's now a faster one, but at the time it didn't produce DNA of high enough quality. So we went with this one, which actually took 4.2 hours. So if we put the scores on the doors here versus our world record, 19.5 hours, 4.2 for Matthew's genome. Then we move to the sequencing part. So the Promethean is loaded. All of 48 flow cells are loaded. 200 gigabases of data, 63x, N50 at 22 kilobases. I think most of you are probably familiar with those kind of numbers. Putting that score up on the door, we get to 6.9 hours. Still some time to go. The biggest change we made was to overlap the compute with the sequencing. So we basically start to compute on little packets of data as they come off the sequencer. And the dry lab portion, of course, includes base calling and alignment and variant calling. And essentially, we optimized this so that the variant calling, which was deep variant from Google, was done on GPUs. The alignment was done on CPUs. And so we basically were just getting the right compute for the right algorithm, and then did it kind of in real time. So that added 3.6 hours, but it overlaps mostly with the sequencing. So it actually often was only like 30 minutes more. So that took us up to 10.5 hours, and we now have a full variant call file at the whole genome level. So then it's filtering, which is reasonably quick, 4.5 million variants, down to, in this case, all the way down to 1. We find essentially this duplication in the troponin T gene that is a known cause of dilated cardiomyopathy, and concluded that this was the cause. That took 46 minutes. So at the end, we were able to make his diagnosis in just over 11 hours, and he was one of the patients, in fact, in whom we were able to beat the record. But much more importantly, he was listed as a result of this genetic diagnosis for transplant, and 21 days later, he received his transplant. There's a picture of him there. Notably, we also sent regular genetic testing because this was a research protocol, and it took, I think, 3.5 weeks to come back, after which time he'd already had his transplant, actually. So just to give an indication of, for these intensive care unit patients, how important speed really is. And this is him a few months after his transplant, next to the Promethean machine that sequenced his genome. And in the end, we published a paper, I think, with 12 cases in it. All of them actually in the end were completed faster than that previous 19.5-hour record. And in fact, in the middle, we stopped barcoding for reasons that are not worth going into right now. But basically, the last 5 were much faster, and pretty much routinely now we can do this in under 8 hours. In fact, yeah, the 3 that went after that were all this is the 8-hour mark shown here. So I made a little Moore's law thing, but for speedy genomes, we added ourselves here at the bottom. These are also long-read genomes, and we published, not quite simultaneously, but around the same time, these 3 papers. You're welcome to go and take a look if you have some interest in the technology there. Maybe it would surprise you, or maybe not, to know that I actually had more emails with Guinness than I had with the New England Journal of Medicine when we published this work. So we did contact Guinness as well, but they made us jump through very many hoops, which was interesting, and also learned things like it is actually Guinness, like the beer, like the drink from Ireland. It's the same Guinness. It's not some other person called Guinness. And apparently the original Guinness World Record book came about because people were arguing about something in a pub over beer, and they realized they needed a book to go look up to decide who was right, because they didn't have the internet or ChatGPT or Google or whatever. So they formed the Guinness World Record book for that reason, to solve a pub argument. Neither of those things I knew. Anyway, this was, we were happy enough to do this, although this was clearly not the main point. The first time the team came together physically, because this was all COVID, was this time when we actually got this award from Guinness. They send someone who then declares you officially amazing. And then one of our patients actually made this balloon sculpture of DNA, which was really fun. Well, obviously I couldn't conclude without going back to our Ferrari. Now, we talked about how much cheaper it would be if it had gone as cheap as human genome sequencing, but what about how much faster it could be? Because the top speed of this car is 200 miles an hour. If we had made this car as much faster as we had made human genome sequencing, there would be a new top speed, which would be 32 million miles per hour. I was quite happy with that, except that I did calculate what warp speed was, and sadly, this is not as fast as warp speed. So unfortunately, we haven't quite made it. So now, doing it in a small healthcare system is one thing, but doing it across an entire country is another. And I couldn't, of course, give a talk at the Australian Genomics group without highlighting the incredible work that has come from Zernissa Stark and the team here in Australia. This is, really impactful on a global basis. Of course, it's, I'm sure, impactful for the individuals within Australia, but this is really world-leading work on ultra-rapid diagnosis for rare disease. We see on an individual basis in our own healthcare system and in a few other healthcare systems that we have helped set up this kind of technology, but not at the scale that you have achieved this in Australia. And I think it's just one of many arenas in which Australia really has led the world for genomics. And I mentioned, I think in this talk, a handful of the folks. There are so many more of you I know working on this, but a handful of the folks that I've been lucky enough to work with over many years on the international stage on genomics, because it really does take a village. It really does take a globe, in fact, to move this forward. And Bill Gates has this saying that most people overestimate what they can do in 1 year, but underestimate what they can do in 10. And you have to be old enough to have been around for 10 years trying to do something before you realize the truth of what he's saying. I think when we first did this, we thought, well, in a year, everybody's going to have their genome sequenced and the world will be a different place. Now, 10, 15 years later, we really are in a different place. I think this really is a place that has delivered. The genome really has delivered. And I think we're all involved in this arena because we see the impact it has on people's lives. Anyway, I thought I added a new thing just about 20 minutes before the talk. I thought I need a new little cartoon for my conclusion slide. So I went to GPT and said, give me an image of Euan Ashley working in a Stanford lab, and this is what it produced. I was a bit disappointed by the universal gray hair here. This particular person looks quite, might be quite smart, but and then they went a bit too young. I think I showed this to my daughter, who said, "Why is it suggesting you're 30 years old?" Which is definitely not true. Anyway, I will not read out the conclusions, but I've been really excited to be part of this and part of applying these technologies along with the global community to more and more patients. And I'm also really happy to have joined you here virtually today. I have shown you the work of many people and also represented many patients. Tiff was kind enough in my introduction to mention this book. If you're interested in these kind of patient stories and the scientist stories as well, I try to tell them it's in a way that is appealing to, I hope, everyone. I also managed, I had to pass an audition actually to be allowed to do the audiobook. And one of my friends wrote this on Twitter after I announced that to the world. So if you're interested in an indecipherable Scottish accent, then I can provide that as well. This is the latest picture of my lab. They've done a huge amount of the work that I showed here. The Undiagnosed Diseases Network is local here at Stanford, national and international, and it's a huge community. The clinical genomics program that does the pharmacogenomics work and the whole genome backbone is shown here, and the Cardiovascular Consortium is shown here. Thank you so much for the invitation. I'd be happy to answer some questions.
Euan, thank you so much. What a wonderful and amusing presentation. And thank you so much for sharing the experiences and odysseys of the families, like the Miller family. I guess while people are thinking of questions, you know, you mentioned that, you know, in the context of the Miller family, these stories drive us forward. And it's bittersweet for those, you know, many families that have not achieved a diagnosis. You know, you put up the data about the UDN efforts achieving sort of 35% diagnostic rates, and then the amazing 6% sort of cost of the cost, you know, the 94% cost saving. How do we more effectively and systematically gather that evidence to embed, you know, sort of earlier genomic interventions in these diagnostic odysseys? Because, you know, health technology assessment and gathering that evidence is so challenging to change policy and get, you know, sort of the national implementation of these interventions, you know, such as you called out Zonitz's work.
Yeah, no, I think it's such an important question, and I just think, frankly, it has been quite frustrating at the fact that we haven't been able to make the case as clearly as we would like, or at least if we have made the case, it hasn't moved the needle as fast as we would like. And now there's great examples of places where it has, and of course, you're all familiar with your local work. The UK has some successes. There's other countries around the world that have had some more success where an individual political leader has really seen the data, taken it on. I mean, the UDN itself, the Undiagnosed Disease Network, because it is quite clinically focused and isn't all about discovery, it's a lot about discovery, actually was moved around the NIH and had to really find a home. And in the end, actually, an intervention from a single family with the chair of the Appropriations Committee of the Congress had the UDN written into the budget, a line item in the budget of the US Congress. So there are stories like that of course give us all hope, but you really would think that the data would speak for themselves. And part of your question was, you know, what should we do? I think we do need as many formalized cost effectiveness analyses as we can, but because it's extremely cost effective. I mean, the normal standard is if you can save, you know, $40,000 per quality of life year, you know, this is like an order of magnitude, maybe even multiple orders of magnitude better than that, because it's literally saving money. So it is a little frustrating in the US context that it's so hard to get coverage, and that we still have to sometimes argue one to one, like literally calling up the payer and saying, you know, this is going to save you money. And actually I had the privilege, I've worked a bit more with the Obama administration, but we had some contact with the Biden-Harris administration here in the US, who were really interested, actually saw this data at the Office of Science and Technology Policy and realized that this was a way of reducing cost, but also asking the question, how do we get this to underserved populations? And so that's another reason I think the Australian work has been so good for the international community, because it's a demonstration that it can be done at scale. And so I wish there was an easier answer, but I think we have to keep beating the drum, keep getting in front of policymakers and lawmakers and payers, and have them just sort of see the data, because the data speaks for itself.
All right. Quick question. So Michael says, you mentioned in your introduction, mentioned AI in your introduction, and that you were Scottish.
Yes.
So despite the fact that it generated an image of you looking like Doogie Howser, if you have to predict the most impactful application of AI in the context of genomic medicine, sort of in this next horizon, where do you think, like, what would it be?
Yeah, great. That's a great question. And, you know, I think that, as I mentioned, you know, these days, mostly I'm asked to talk about either AI or genomics, but of course they do overlap. And, but they don't overlap as much as I think they will in the future, But right now, when I am giving examples, there's a few more in the last few months where we've literally been using generative large language models in clinical care. Like it listens to the consultation and makes a note for you. I mean, that's actually real now. But a year ago, that wasn't really true. And actually, genomics, and in particular cases like Matthew's, where we use deep variant, the Google's deep learning based model for calling variants, which is, has better data than almost any other variant caller, especially for long reads. That was one of the few places where you could genuinely say, at the moment, AI is affecting clinical care in this way. And we did a review for the New England Journal on, I think, AI and molecular medicine, if anyone's interested. And we had to actually really work quite hard to demonstrate where today in clinical care, this was today, that was now a year ago, but the AI was impacting clinical care, and genomics was the answer. So variant calling is one place. I think that variant classification is clearly another, and I mentioned that briefly, but I think now we've seen with AlphaFold and then Alpha Missense a bit of a sort of excess, so, some more rocket fuel to the fire of how do we classify variants, and especially, of course, variants of unknown significance. So I'd say those are two examples right now, and two very salient and real examples in genomics. Slowly, slowly in AI terms, but rapidly in any other kind of speed, which is to say month by month, there are now examples accumulating in the rest of the healthcare system. But I'd say for much of the last couple of years, the only place that AI was really impacting healthcare was in variant calling in genomics. As opposed to being an interesting research,
Yes.
so it can classify chest x-rays, great, but how many people were actually using it that way in clinical practice? That is changing, but in the last couple of years, actually, genomics has been one of the few places that AI has actually been there.
Yeah, and it's causing interesting for health policymakers. It's an interesting landscape that they have to navigate at speed because it is being taken up so quickly. So David Thorburns asked a question. He would know everything about the MECA pathway that is affecting the Miller family. So he is asking a question about what are your thoughts on the status of ultra-rapid or rapid functional genomic testing? For example, calmodulin seems to be expressed at high levels in PBMC. And our colleague David Stroud in Melbourne University has shown the feasibility of PBMC proteomics in a 3-day turnaround time.
Yeah, I mean, I think that one of the things that, so there are a number of things that we've been sort of looking to have happen for some time and never been quite sure when it would sort of break through into the mainstream as opposed to being something we can do in the research lab. I would say RNA is one of them, and transcriptomics, which we regularly use in the UDN, but almost never use clinically, though maybe we should. But again, it's a question of who's paying. And then I think other functional genomics measures, if they don't all take 8 years, I showed that one example with a dissection at a mechanistic level that took 8 years, like that we don't have time to wait for that. But there are quite a few assays, and that's a great example that he brings up there of a proteomics assay that could help within the course of just 2 or 3 days. I mean, most of the conditions don't need a 12-hour, 8-hour turnaround. Of course, there are plenty of situations that do, but often a one week sort of turnaround can be really, first of all, very cost effective, and very meaningful. Yeah. And so I do think we should explore more and try and pick from our sort of panoply of potential functional genomics assays. We could really, I think, push that number up. I mean, 30-40% solve rate is, that's great compared to 0, which is where many of these patients start, but it's not good in the grand scheme. We really want to be pushing that up, and there are ways to do that. I think the long reads will help. I think thinking about methylation may help a bit. Being able to do structural variants will help a bit. Functional genomics, RNA, definitely 2 significant areas where we could push that number up further.
So Euan, I'm going to have to apologise to those others who've asked questions. I'm sure we could keep chatting for a while, but we do need to wrap up. Thank you again. It's been really wonderful having you join us this morning or this afternoon.
Oh, my pleasure. Yeah, thanks so much for the invite. Great to be there. Thanks, Euan. Take care.
And thank you all for joining us for the last DNA Dialogue for 2024. I'd particularly like to thank Dorothy, Liana, Ellie and Will for their efforts in bringing this wonderful series to us, and all our speakers for their contributions this year. Go and buy Euan's book and also keep your eyes peeled for news of the 2025 DNA Dialogue series, which will be kicking off in February. Thanks very much for joining us.